Hi, my name is Jeff Petracca, and I'm an educator and entomologist here at the DNA Learning Center at Cold Spring Harbor Laboratory. Welcome to part 2 of our Genetically Modified Organisms Lab. If you're just joining us, I would encourage you to go back and watch part 1 of this experiment, just so you are caught up to speed on what we are going to be doing here today. And I wanted to remind you that this lab is particularly close to my heart as someone who studies insects, which have a very close relationship to plants. And we are essentially looking at the genetic modification of various different plants that are incorporated into snack food products. And in particular, we're looking at a cheesy corn chip as well as an organic corn chip. Okay, so let's take a look up at the board just to review what we've done already. Okay, so just as a reminder, the purpose of our lab here overall for both part 1 and part 2 is to determine if various snack foods, so these organic corn chip and this cheesy corn chip specifically, contain evidence or contain components that show evidence of genetic modification. And so the sort of metric that we're using to assess that is 35S. So this is that promoter that I mentioned last time from the cauliflower mosaic virus that genetic engineers will incorporate with their gene of interest that they're trying to put into a host organism in order to guarantee or make sure that the expression of that gene of interest goes forward. And so that's what we're using to detect this genetic modification. And because we're dealing with plants here, we want some way of knowing whether or not we've successfully amplified DNA from these food products at all. And so a good way to, a good control that we could use to do that is a ubiquitous plant protein. And we're using the protein tubulin. Okay, so this is serving as our control for this experiment here. Okay, so that's our purpose, and that's what we set out to do. And so remember that last time what we did was we isolated DNA from both of our snack food products as well as a positive and negative control. And the positive and negative controls are, again, what those are doing are they're verifying that the primers that we're using are actually successful. So remember, like I said last time, this is one of our most challenging labs just because it's hard to extract DNA from processed foods, you know? So we want to make sure that if we don't see any results, the reason that we're not seeing any results is because the extraction from the snack foods were not successful, and it's not simply because I used primers that were not good. Okay, so that's the reason we're doing that positive and negative control for, and then we used, of course, GMO corn and non-GMO corn for that. Then we amplified DNA by PCR, and we did 2 different amplifications. We did that tubulin amplification, and we did 35S from that same stock tube of purified DNA that we extracted and isolated. Okay, so for today, what we're going to be doing is we are going to analyze the PCR product. So those have been running. It took about an hour and 15 minutes or so, and that's all done now. And we're going to analyze whatever comes out of that PCR, the amplicon specifically, that's the same as saying PCR products, using a technique known as gel electrophoresis. And so if you've watched the DNA Learning Center videos, you'll know that gel electrophoresis is a technique that separates out molecules on the basis of their molecular weight. And when we're talking about DNA, it separates them out on the basis of size, or more specifically, the number of base pairs that they consist of. And so that'll allow us to visualize the amplification that we're getting from our PCR. But in order to figure out what we expect to see, or to verify that we're actually amplifying 35S or tubulin, we need to figure out approximately how big they are. And that's where the bioinformatics comes in. So we're going to be using a couple of computer exercises to figure out what the size of tubulin and what the size of 35S should be. And the whole goal here, again, is just to see how big they are so that we can actually compare that to our gel results and verify that, yes, indeed, we do have tubulin and 35S. Now, to put this all in context, just as a reminder, last time we talked about genetic modification using the Bt gene. So that was that bacterial toxin that kills off insect larvae. And so I wanted to use another example here in the form of a herbicide resistance gene. Okay, and to be clear, when scientists develop these genetic modifications, there are a number of ways to insert them into a host organism, but one of the common ways to do this is to build a recombinant plasmid. And so here we have an example of a recombinant plasmid. Remember, it's a little tiny ring of DNA. And so we have that gene of interest over here. And so that could be like your BT gene or your HT gene, for instance. That's your bacterial toxin or your herbicide tolerant gene. And that 35S promoter that originated from the cauliflower mosaic virus, that's placed right before our gene of interest, just to guarantee or ensure the expression of that gene. And there are a couple of other things here. There's a couple of other things that I don't think I really highlighted last time. But what you want to have on a recombinant plasmid in particular is an origin of replication and some selective factor that we can use to assess whether or not our transformation or the insertion of this particular recombinant plasmid was successful into our host cells. And so here they're using antibiotic resistance. And antibiotic resistance is just a way to select for those cells that have that genetically modified element. And the origin of replication is meant to basically replicate this plasmid again and again and again. You want to make sure that this plasmid gets replicated and passed down to other cells in the organism. You don't want to just incorporate this recombinant DNA into one cell and be done with it, because that cell, that's not going to really help to create a genetically modified crop or whatever. So that's what that origin of replication is supposed to be. And so let's say that let's talk about this herbicide tolerant gene for a moment. And so we talked about last time a little bit why a farmer might want to use a herbicide resistant crop, or why that might be a desirable thing for them to have. And so imagine that you are the owner of a major agricultural firm with acres and acres and acres of corn crop. And you want to ensure that you maximize the amount of yield that your crop produces, and obviously you want to make sure that your plants are healthy and growing as they should, or as best as they could. And weeds, unfortunately, are plants that will start cropping up anywhere. They'll crop up in a major crop, they'll crop up in a garden, they'll appear pretty much anywhere. Right? So they're going to be sapping energy and nutrients and water away from your crop. So therefore, you would behoove you to kind of get rid of them, right? But a lot of herbicides, which are chemicals that are used to kill weeds or other plants, are generalists. They're going to be broad spectrum. They're going to affect a lot of things. So if you were to spray an herbicide on your corn crop, you might accidentally kill your corn plants, right? So you don't want to do that. But what if you could incorporate a genetic element into your corn plant, or your corn crop, I should say, that is able to resist some of these broad spectrum herbicides? And that's exactly what this HT gene, or these herbicide tolerant genes, do. And so one of them is a glyphosate resistance gene. And so this component, glyphosate, is an element within the generalist herbicide Roundup. And so what this generalist herbicide will do is it will kill. It's used in a lot of different applications, and it will kill broad spectrum, just all plant material. So I know it was previously used for driveways and walkways and things like that, to kill weeds that crop up in between the pavement. It pretty much kills everything. So this glyphosate resistance gene can be incorporated into a little bacterium, and then can be transferred into the plant cell, into a corn crop, let's say. And voila, there you go. Now, all of a sudden, your corn crop will be resistant to this glyphosate element, allowing you to spray Roundup on your plants, and it will not harm them, but it will kill those weeds that are taking away from the health of your crop. And so that's the idea. And I wanted to remind you guys that this genetic modification does not necessarily have these nefarious purposes that it seems to be portrayed as in a lot of popular culture. And so there are reasons why this is important. And it's not just to line the pockets of big corporate types all the time, right? The idea is to make farming and the agricultural industry a little bit more efficient and more technologically savvy. So we're using technology here in order to enhance our products. Okay, so we'll talk more about the pros and cons of GMOs later, but for now, just I wanted to put this in your mind and just kind of run you up to speed on where we're at. Okay, so now for today, what we need to do is analyze the PCR products by gel electrophoresis. And when we talk about gels, at the DNA Learning Center, we typically do agarose gels with our students. And so today we're using a 2% agarose gel. Okay, and there's a component in here also known as gel red, just so you guys know. And gel red is a DNA stain that will allow us to visualize DNA wherever it may find itself in our agarose gel. Now, agarose is a sugar originating from a seaweed. And so the way that gel electrophoresis works is the larger molecules of DNA or of whatever molecule you're loading into the gel will have a much more difficult time navigating through a polysaccharide matrix of this agarose compound. Okay, so the higher the larger the size of that molecule, it will not have an easy time at getting through the gel. And so it will not travel quite as far, whereas little pieces are going to travel much further. And so that's how these gels work. And now for today's experiment, we're using a very specific sort of loading style here because we have a lot of stuff going on. So I wanted to go through it with you before we actually do it in person, just so you're clear on what you're seeing. And so if you pay attention over here on the board, you'll notice that we have a gel looking directly down on it. And we've got two rows of what we call wells. And so I already went ahead and poured this gel for you, but essentially that entails me just heating up agarose of a specific concentration and then pouring it into a casting tray, which I'm going to show you momentarily. And I put two combs in the gel. So those combs leave behind depressions as the gel hardens or cures. And we call this, therefore, a double combed gel. And we're using this double combed gel just to increase the number of wells that we have because we have so many samples going on here. Okay, so remember that we have both a tubulin and a 35S amplification for our sample. So we need to provide space for both of those. And so up here on the board, you can imagine that number 1 here might correspond to your positive control. Number 2 might be your negative control. Number 3 might be the organic corn chip, which if you remember, I called that C in our extractions last time. Number 4 down here might be the cheesy corn chip. And if you remember, I called that D in our extractions. Okay, and each one of them have their own tubulin and 35S components. So we need to essentially load both of those. And we're going to do those side by side just so that we can get a little bit better of a visualization of the samples. Now, if you notice, the first lane in both of, the first well rather, in both of these rows of wells has an L above it. And so what that is a ladder. It's a reference marker that we're going to use to basically compare that to the size of the fragments that we see in the tubulin and 35S amplifications for each of our samples. And so this ladder that we're using is specifically called a 100 base pair ladder. And that 100 base pair ladder is basically a mixture of DNA that will separate itself out such that every band that we see on the gel is 100 base pairs different in size. So for instance, it's going to go 1,000 base pairs, 900, 800, 700, 600, 500, 400, 300, 200, and 100. So that's what you're going to see, like a banding pattern where every band represents 100 base pairs different. So the largest ones, the 1,000 base pair ones, are going to be up here, and 100 are going to be down here, reflecting that differential migration rate of DNA of varying sizes. So the smaller ones will go further than the larger ones. And so that's the plan. And I'm going to go ahead and load our gel. And I want to show you how to do that. So let's jump over to that. Okay, so let's go ahead and load our gel. So as I mentioned before, I have already poured your 2% agarose gel right here. This is a double comb gel because I have 2 white combs here that provide 2 rows of wells for me to go ahead and load samples into. And essentially pouring agarose gel is just like making fancy science jello. So this is a casting tray right here. These little blue things are little end protectors that will essentially provide a place for the gel to sit in and harden. And I'm going to go ahead and pull those off because it's been sitting here for about 15 minutes now. So the gel is already hardened, or I should say cured, and it's ready to be used. So I'm going to pull these combs directly up and out of the gel here, like so. Okay, and what I have left behind here is very much like the image that I showed you on the board momentarily a little while ago. So I have a row of wells right up here at this black line and a row of wells down here at the red line. And so now those the black and red lines, those correspond conventionally to what's known as the anode and the cathode. So the negatively charged and positively charged electrodes in a gel chamber, in a gel electrophoresis chamber rather. And so if you notice, I actually have a gel electrophoresis chamber right here. And if you take a look on the box here, you'll notice that it has a black and red line on it as well. It also has a black and red wire. Those are corresponding to the negative and positive ends of the chamber as well. So I'm going to take this casting tray, gel and all, and I'm going to drop it right into the gel electrophoresis chamber, matching the black line to the black line and the red line to the red line. Then I'm going to take some buffer, because essentially the way that gel electrophoresis works is it uses electricity to pull molecules, charged molecules, through this 2% agarose gel. And basically, the bigger the molecule, the harder the time it's going to have navigating through the polysaccharide matrix that is this agarose gel. Remember, agarose is a sugar, right? So when I poured that hot agarose, it cured in such a way to make this matrix of sugar molecules that basically provides a resistance to the flow of molecules through it. So the larger the molecule, the harder time it's going to have getting through the gel, and the smaller the molecule, the easier time it's going to have getting through that polysaccharide matrix, and it's going to go a little bit further in the gel. And that's basically how gel electrophoresis works, more or less. The only problem is I don't have a conductor. So if I were to just plug this box into a power supply right now and run current through it, it's not going to really be conducting electricity very well. So I want to make sure that I use a conductive material in the box to pour over the gel, and that's what this is. So this is a buffer, and this buffer, which buffers are solutions of ions. Basically, that's all you really need to know at this point. Buffer is basically a solution of ions, meaning it's going to conduct the flow of electricity. So I'm going to go ahead and pour my buffer on one side of the gel here, and I'm going to pour it on the other side as well. And that's going to basically pour just over the top of the surface of the gel completely. So I want to cover the gel just enough. I don't want to put too much buffer in there. I want to put just enough to cover the surface of the gel. And the reason for that is because it's harder to load if there's too much buffer in the box, and your sample might float up and out of those wells. Okay, while we're talking about electricity and current here, I'm going to go ahead and just get my power supply plugged in here. So I'm going to take the lid here, this black electrode. This is my power supply that will provide the electric current that we need to run samples through the gel. So I'm going to plug my black electrode into one of the ports here that is indicated with a black color, and I'm going to plug my cathode, my red electrode, into a red port. Okay, and so we're good to go on that end. We'll turn the power supply on momentarily. So now, the plan here is to load 18 microliters of sample into each one of the respective wells. And I'm going to keep it consistent like I showed you up on the board. I'm going to do for each sample tubulin first, then 35S, then tubulin first, then 35S. For the organic corn chip, the cheesy corn chip, then the positive and the negative controls. Okay, so let me pull those out of our thermal cycler over here. So these guys have been sitting here now and overnight. And if you recall, I had my positive and negative control here in the first tubes in this little strip here, and I have the clear hinge tubes representing the tubulin and the black hinge tubes representing the 35S. If you come here to do this lab at the Learning Center, we do that the same way. So clear hinge is supposed to be tubulin and 35S is the black hinge tube. So I'm loading 18 microliters, meaning I'm going to be using my yellow pipette. I'm going to set that for 0180. Okay, and I'm going to put the positive and negative controls on the bottom lane, on the bottom row of wells, and I'm going to put the organic corn chip and the cheesy corn chip up on the top row of wells here. So since I'm starting here with my positive control, I'm going to go to my first stop, and I'm going to take up 18 microliters of liquid. Now, when you're loading a gel, you always want to make sure your hand is stable when you're putting the sample into the well. You don't want to be going crazy all around here because you might actually damage the well. You might misplace where you're putting the sample. So when we do this, when I do this anyway, I like to put my elbow on the table. I have to stand when I do this, and I'm going to use my other hand to guide the tip of the pipette into the well. And you're essentially, if you pour just enough buffer into the box here, you're basically just going right below the surface of the buffer, and you're aiming right for whatever well you're trying to load into, and then you should be there theoretically. So I'm going to go down here to the second row. I'm going to go to the second well in that row, because remember, we're putting the ladder in the first well, right? And I'm going to go right underneath the surface of the buffer. If I move the tip of the pipette up and down, basically perpendicular to me like this, you'll see the gel will move back and forth. That's good. That means we're there. So I can go ahead and deposit my sample. When I do that, I am going to only push to my first stop. I don't want to go to my second stop now because I will blast the sample out of the well and it'll float away into the buffer. That's not good. We don't want that, right? So also, I'm keeping my thumb on this button the whole time as I'm loading the gel. If I let go, as many students have a tendency to do this, by the way, if I let go when I'm in the well after I deposit my sample, I'm going to take my sample back up and I'm going to take buffer along with it. We don't want to do that. So just have confidence that if you push down to the first stop in the well, the sample is there. And as a matter of fact, you can actually see the sample, and it's hard to see on the camera here, but you can actually see the sample sitting in the well. It's a little purple rectangle. Those wells are rectangles, right? So this PCR mix actually has a little dye in it that is pink for that very reason. So when we are loading a gel, that pink color will show us that we've actually successfully deposited the sample in the well, pink or purple, whatever. Okay, so now I'm going to go ahead and load the 35S for the positive control. And so I go to my first black hinge tube, that's my positive control. And I'm going to deposit that right next to the tubulin sample that I just loaded. So I'm going to go right in there, and I'm going to go to my first stop. Just like that. And I deposit my sample. Again, I'm changing my tips just to make sure that I am not contaminating my sample. At this point, if we mix these things together, these tubes together, we're not going to be able to tell much of anything because the samples will have been mixed. So next, I'm going to go to my tubulin tubulin negative control. I'm going to put that right next to the tubulin, to the 35S for the positive control. Then I'm going to go to my 35S amplification for the negative control, and I'm going to put that right next to the tubulin. Okay, so there we go. Those are my first samples are done. Let's go ahead and proceed to the organic and the cheesy corn chip. I think I'm going to I'll do the cheesy corn chip first, actually. So I'm going to put those two up on the top row here, skipping that first well again. So I do my tubulin, I do my 35S right next door. And last but not least, I have my organic corn chip. So here's my tubulin, and here's my 35S. Okay. All done. So now that these have all been loaded into the gel, this is garbage. I'm going to throw the rest of that away. We don't need that anymore. But before we get the gel up and running, I need to load ladder. Now, the ladder is a little bit more concentrated than the other samples. So what I'm going to do is only load 6 microliters of ladder. I'm not going to put that full 18 microliters just because the ladder is going to light up really, really brightly, and you're not going to be able to really discern the different bands that we want to see. So I have that 100 base pair ladder right here, and I'm going to put 6 microliters into the first well of each row of wells in my double comb gel here. Oh, I didn't have to actually switch the tips, but that's okay. Better safe than sorry. Okay, and 6 microliters. Okay, all done. So now, that means that our gel is ready to go. It's all loaded up. So I'm going to slide the lid onto the electrodes here, double checking that the black matches the black and the red matches the red. If it's reversed, your gel is going to run backwards and your samples are going to float away. Make sure you're in the right orientation at this point. Otherwise, you will have wasted all your time. Now I'm going to go over to my power supply, and I'm going to turn that on over here. You'll notice that there's a little 0 up on the box here. So I'm going to press the up arrows until we get to 130 volts. Okay, then I'm going to push this little button and I'm going to set that for 30 minutes. So we're going to let that sit and run for 30 minutes. Then I press the little running man and away it goes. Our gel is running. Okay, so we'll check back on this in about 30 minutes. But in the meantime, let's talk about what we expect to see on our results. So in order to understand what we expect to see on our electrophoresis, or for our gel results, I should say, we need to gain a better understanding of how big the 35S and tubulin amplicons are supposed to be. Now, up till now, we really haven't talked much about this, right? I haven't actually told you how big those gene regions are at all. We have no idea. All we know is that if we loaded a ladder over here in the first well, that's going to separate itself out such that every band is different from the last by about 100 base pairs. So here we have 1,000, 900, 800, 700, 600, 500, 400, 300, 200, 100, right? So let's say that I loaded my positive control samples right next door here, the tubulin and the 35S. And then right next to that, I put my negative controls, tubulin and 35S. Okay, and remember, this is the presence of GM and the absence of GM, genetic modification. So what do we expect to see? Well, I don't know, because we need to figure out the size of those amplicons. Now, how are we going to do that, though? Well, it turns out that we can use bioinformatics to help us answer that question. So what I can tell you is the sequences of the primers that we used to prepare our PCR reactions, our amplification reactions. Okay, so those primers are actually located in the laboratory protocol, and then believe that laboratory protocol is actually attached to this video in the resources section on DNALC Live. Okay, so we actually have access to the primer sequences. And remember, primers are very short sequences of bases. You know, these are very specific. I told you I wasn't going to go too deep into primers, but it is important to understand what primers are. They're very specific, and they bind both upstream and downstream of a particular gene region of interest. So here's my 35S, for instance. I'm going to have one little baby primer. It's about 20 to 30 base pairs in length, binding upstream, and one that binds downstream on the complementary strand. And during PCR, they amplify like so. Okay, so those primers signal to DNA polymerase where to initiate replication and basically begin adding nucleotides. And so theoretically then, if we could figure out where those primers are supposed to bind in the genome, then we can figure out how big our amplicon is supposed to be. This whole thing is going to be our amplicon, right? And again, that's basically just what was supposed to be coming out of our PCR. right? So we could basically figure out the size of each one of our gene regions. But where do we get that information from? How do we know where it binds? Well, we could compare it to a database, to a sequence database. And it just so happens that there are such sequence databases out there. And so we are going to perform what's called an electronic PCR. And we're going to be using a sequence database known as GenBank. It's actually the National Institute of Health's sequence database. And so what these databases are is basically a collection of sequence information. And when I say collection, I'm thinking in terms of like an insect collection or a herbarium collection where they store a bunch of plant specimens, or even a living botanical garden. And so this just is a collection that is virtual, and it contains sequence information. So if we can essentially compare our primer sequences to this database, we can see where those primer sequences will match in such a database, in GenBank. Okay, so let's do that. Let's actually first go to pull our primer sequences. And so if you guys go to our resources tab on DNALC Live, you can pull up the student protocol for this laboratory that was attached under the resource section. So here is our student laboratory protocol. And what you can do is scroll all the way down, and I believe it's on page 9 of the PDF document here. There we go. Right over here, we actually have access to the primer set. So the forward, the upstream, and the reverse, the downstream primers that were used for both tubulin and 35S. Now, it doesn't tell you which one of these is which, however. So we have to figure that out. Okay, so what I'm going to do first is I'm going to go open up a new tab, and I'm going to go to GenBank. So you guys can just simply type in GenBank into like a Google search or something, and this is what's going to pop up. Okay, so once you're on GenBank, you can go ahead and click this little pull down menu right at the top here, and then you're going to click this tab here that says BLAST. And that brings you up to this page here. So BLAST stands for Basic Local Alignment Search Tool, right up here. Okay, so BLAST essentially is a search mechanism that will search the entirety of the GenBank sequence collection and align whatever input that you put into the search tool with all of those sequences to come up with a match. So we're going to be doing a nucleotide blast. So if you click on that, that'll open up this page here, and you'll see all kinds of like search criteria and filters and things like that. But we're primarily interested in this box right up here. So where it says enter accession numbers, GIs, or FASTA sequences. Okay, so go back to that protocol that we were looking at a moment ago, and you're going to just copy and paste one of the primer sets. Take the whole thing. So I'm actually going to go from the nucleotides here straight on down to the other primer set right here. Okay, now I'm going to just control copy and then go over here and paste it, control V, into the query search box up here. Okay, so what you have left over here, you have your primer set, your forward and reverse primer, and we have this random stuff that we want to get rid of. That's basically indicating the polarity of the primer sequences. We're going to get rid of that though, because when it says up here, enter accession numbers, GIs, or FASTA sequence, it wants you to put whatever sequence you're inputting in a particular format, in particular FASTA sequence format. So basically just the bases, and that's it. We don't want anything else in there. So you're going to get rid of everything that's not nucleotides, that are not bases. Okay, so once you have that input it into the query search, this now becomes your query. So this is the thing that you're comparing to all of the entries in the database. Okay, so if you scroll down a little bit, we're not going to mess with any of the other filters except for this one where it says program selection optimized for. Now, it is defaulting when the first time you access the BLAST database, the GenBank database here, it defaults to highly similar sequences. However, because we are dealing with these primers here, which are very small, remember, they're only like 20-something base pairs in length. So we don't want to There are very small pieces, there's very small fragments of DNA. We want to click somewhat similar sequences because otherwise it will come up with too many matches and it will not provide any useful information. So you're doing somewhat similar sequences down here, okay? Then you're going to click blast. And so what that's going to do is run your query, run your search tool, or run your primer set against everything that's in the GenBank database. Okay, so it takes a few seconds, sometimes longer than others, depending on, I guess, how many people are searching at the same time. And what you have is, once this page comes up, is your results, your matches to your query. Okay, so what we have here is a bunch of things. We have, if you scroll down a little bit, you'll see all of this stuff. And basically, these are all entries in the GenBank database that your sequence that you inputted, your query, is coming up with a match for. Okay, so there's a lot of stuff here, and there's a lot of stuff that may look very unfamiliar to you. A lot of stuff, I mean, I don't know what half of this stuff is either. A lot of these are cloning plasmids and things like that. That's what that little P stands for, and this entry here. But a lot of this, we're not really interested in this, right? We are doing an experiment regarding 35S and tubulin, correct? So that's what we're looking for. So what you're going to want to do is basically look for any entries here that match, that have those phrases in them. And if you scroll down a little bit, you'll notice that there is a cloning vector, and lo and behold, it says 35S. Interesting, right? So this is the one we're going to actually be focusing on. Before we do that, though, I want to talk about all these other numbers and things that are going on over here. So first off, like I said before, GenBank is a collection. It's a virtual collection of sequence information. And so like another collection, there are record numbers that essentially are associated with all of the sequences in their virtual collection. And that's what this is over here. So this accession number. So each one of these seemingly random numbers all the way over here on the right are called accession numbers. And those accession numbers are the unique record number that is associated with the sequence that's been published to this database. So scientists from around the country and around the world can go ahead and publish sequence information to this database, and they're given an accession number. Okay, so if I were to click one of these random accession numbers, so let's click this guy right here. It's another 35S actually. Okay, so this will basically bring up the information surrounding that particular sequence. And so we have the size of the fragment that is in the database here, whether it's DNA or RNA or whatever. It gives you a little bit of information about the authors, whoever published it, how it was published, if it was associated with a particular plant, with a particular journal or scientific publication, and it gives you the sequence itself. Okay. It even gives you the translated sequence, and that's right up here. And so this basically represents the letter codes for the amino acid sequence that corresponds to the protein coding element of whatever sequence has been published. Okay, it gives you the year that it was published as well. So basically my point is there's information that can be had when you click, that can be figured out when you click the accession number. And so I'm not going to go much more into that, but for now, if you look to the left of the accession number, you'll see this % identity. And this is like a % match, essentially. So basically, what this is saying is it tells you how much of your query matches the subject sequence. So according to this, every single one of these is 100% match. That means that the query that we input it into our BLAST search matches each of these 100%. Okay, so there's no mismatches going on. Okay, so that's not very telling because all of these are 100%, but that's okay. That, again, it's a short primer sequence. We would expect it to possibly be present in many different locations. Then we have our E-value. Okay, so our E-value is kind of like a p-value from statistics. And basically, the lower the E-value, the more significant the result. And so if we have a really low E-value, that means that the probability of having this match having occurred by random chance is pretty low. Okay, so basically working very much the same way as a p-value would. Query cover tells you that 100% of your entry, your query, matches the particular subject that is indicated. And these are metric scores that tell you how good of a match your your query subject matches are. And I'm not, to be honest with you, sure how GenBank, the algorithms that GenBank uses to calculate these, but for all intents and purposes, we're not really interested in these at the moment. We're basically just interested in figuring out the size of our particular sequence, of our particular amplicons rather. So let's go to this one here, and you can see it's in purple already because I've clicked this one already. So I'm going to click this entry. Okay, so this brings up a different page. So before, remember, I clicked the accession number and it brought up information about the author and whatnot. This simply brings up what's called an alignment. Okay, so an alignment is basically a side-by-side comparison of your query compared to where it matches in the particular subject in the database. Now, if you notice here, you'll see that there are quite a few matches. Okay, so we've got one here. So so if you look at each one of these, we have it actually matching multiple different places in this particular entry. Okay, which is going to be a little confusing perhaps, but we'll talk about exactly what's going on momentarily. But for now, just understand that that's what this is representing. So we have a match for our query. This is one of the primers. Right? I believe that's the reverse primer matching to a location in the subject. We have the reverse primer again matching to a subject in the to a region in the subject sequence. We have the forward primer binding here, and we have, I guess, the forward primer again and the forward primer again. So we have, point is we have multiple locations that our primers are binding in this particular match. Okay. Now, if we scroll down a little bit, here's the next one in that list, and we see that 35S is listed again. And to be honest with you, I like this one a little bit better because there are fewer matching locations. So why don't we work with this one? Okay, the other one's a little bit more confusing, and it's going to get a little math heavy. So let's take a look at this one. Okay, so what it's showing you is where the forward primer binds and where the reverse primer binds. Okay, so notice I have two forward primers here. Okay, and I have one reverse primer. Interesting, right? Well, let's see where they bind. So it turns out that we have these numbers here that correspond to positions, nucleotide positions in the subject. So that's what these bigger numbers are. So remember, this is a sequence that has a length of about 5,355 base pairs. The subject, on the other hand, that's where, the primers, on the other hand, are only about 50 base pairs in length. So here we have the positions of each one of those itty bitty primers in comparison to that large sequence. So basically, our primers are binding at a position of 1309 to 1285 and 821 to 844 and 1148 to 1171, right? So basically, these are corresponding to positions. How can this help us to figure out the size of our particular amplicon? Well, let's take a moment and figure out how these numbers can help us. Okay, so to help us figure out the size of our amplicons, what we're going to do is build ourselves a picture here. So working off of the ranges in the match that we have in GenBank on the left-hand side of the screen here, we're just going to indicate where exactly our primers are binding in the subject. Okay, so let's start with the smallest piece. So the smallest range is 821 to 844. So basically at the 821st position to the 844th position, that's where one of our forward primers, we put an F down there for forward, are binding. Okay? Then we go to the next range, and we have 1148 rather to 1171, right? And that's another forward primer. Then we have, last but not least, the reverse primer binding all the way up at, what is it, 1285 to 1309. Okay, I'll put a little R down there for reverse. So what's the deal with this? How are we going to basically use this to figure out the size of 35S? Well, let's start with the smallest piece first because we have forward to reverse, correct? So this basically reflects our 35S primer region, our amplicon that we should be getting from our PCR. Why, you might ask? Because if you remember, the match that we have in GenBank on the left over here has 35S in the name, so therefore that is likely going to be a 35S 35S primer set that we'd searched for in GenBank, since that was the match that we came up with. Okay, so basically what that's saying is that 35S is in here somewhere. Now, to get the size of our amplicon, now what is the amplicon exactly? Well, it turns out the amplicon should be from the beginning of the forward primer all the way to the end of the reverse primer. So therefore, this whole bit here is our amplicon. So to get the size, we just simply do 1309 minus 1148. So the biggest number minus the smallest number in our match here, and we get 161. But that's not quite the size of the amplicon. That is these positions, these numbers here are referring to positions within our subject, right? So they are indexes essentially. So we need to add one more to get the actual size. So we get 162 base pairs in total. That's the size of our 35S amplicon. Okay? So if you think about it, like if you think about 3 people standing in line, for instance, just to understand where I'm getting that from. So we have 3 people standing in line. We have 1, 2, 3. The second person is obviously second in line, but he is technically, you know, the first person after the original, after the one that's standing in the first place, right? So it's basically the same principle. That's why we're adding that one extra base pair to come up with the total size. But now, what's going on with our other region over here? Well, we have the forward primer, and that goes all the way over to the reverse, believe it or not. So we have this big, big amplicon as well. So now, to get the size of this one, we're doing 1309, same thing, biggest minus smallest, so minus 821. And we come up with 488. Okay, and then just like before, we're going to add one more base pair, and we come up with 489. Okay, so this is, in this particular example on GenBank here, we're getting 2 amplicons. And so what this is indicative of, it's possible that there was a second 35S region promoter that was incorporated before the first one, so before the original. So sometimes you have them being incorporated back to back. Sometimes you have them being incorporated a bit apart from one another. And so that's probably the case here. So notice that I can tell you for a fact that the size of our 35S amplicon is supposed to be 162. But this 489 just simply reflects that there's a lot of gene region between this first promoter here and this second one over here. Okay, so where that second one starts. I'm sorry, over here, where the primer actually starts. So there's some flanking region right here that we don't. That's making that size a little bit bigger. Okay, so that's why we're getting those 2 amplifications. So on a gel, we'd be seeing 2 different bands for our amplification because we're getting multiple different amplicons for 35S. Okay, so now by process of elimination, therefore, tubulin is going to be a primer set that was located on that protocol. Okay, now for time's sake, I'm not going to go through the same process with you. I'm simply going to tell you that the size of that amplicon is about 187, 192 base pairs in length. So it's right around 190 base pairs in length. Okay, so let's take a look at what we should expect to see on our gel. Okay, so here is our gel picture that we started earlier. So remember, we have our ladder here in the first well here, the first lane, and we see it separated out. So we have 1,000 base pairs up top here, and we have 100 base pairs down here. Okay, now we just came up with the size of each one of these amplicons. So we have 162. Let's just assume that we have 1 amplicon. So again, there's possibilities that there may be multiple amplicons for 35S, but let's just assume 35S is 162 base pairs, just a single amplicon. Okay. Then for tubulin, we came up with, or I told you, the size is going to be about 187. So I've done it on GenBank, and it's worked out for me to be about 192 base pairs in some cases. So if that's the case for you, approximately 187. Okay, so now what should we expect to see then for our results here? Okay, so if we talk about our positive control, our control that contained genetically modified corn so that we know that there is genetic modification present, what should we see for tubulin? So it turns out we should have one amplicon, right? So one amplicon, that is 187 base pairs in length. So what we're going to see is right over here, right up where the 200 mark is on our 100 base pair ladder, we should see one band, okay? A little bit below the 200 mark, the way that I drew that. It was a little bit basically in line with the 200. I'm doing it at an angle here. Sorry about that. So there you go. A little bit below the 200 base pair mark on the ladder, okay? Now what about the 35S? So if we're assuming that there's only one amplicon, then yes, there should be one band just a little bit below where the tubulin is. It's almost like a little step, a little mini step. Okay, so those are our results for our positive control. Okay, so basically, that's telling us that if we get amplification for that 35S PCR, that means that yes, there was some sort of genetic modification going on involving 35S promoters, okay? Now, what about for the minus control, the negative control? That was, again, wild-type corn that had no genetic modification. So should we see tubulin amplification? Yes, and we should see it at basically the same level as we did in the positive control. So right around the 200 base pair mark on our ladder, okay? And then what about for our 35S? Nothing. There should be nothing here. It should just be blank. There should be no banding whatsoever because there's no 35S present. No 35S has been incorporated into this wild type corn, so therefore no genetic modification has taken place. That's the idea. And we extend that to our food products as well because that will tell us whether or not we have genetic modification present in our food products, and we should be able to interpret the results based on that. Okay, so in all of that time, our gel has actually finished running. So I'm going to go ahead and turn off the power supply on my gel chamber. I'm going to open up my gel box. And now, I told you earlier we added gel red to our gel here, to our agarose gel, right? So we can't actually see our samples at this point. That pink color contained a dye that shows us that the gel actually ran. So you should be able to see these little pink blobs on the gel, which actually you're not able to see because it's since diffused away. I took a little bit of a breather in between here, so it's since diffused away. But we should still be able to see them up on the UV illuminator. That gel red will wind up intercalating in between the bases of DNA. So basically, wherever it encounters DNA in the sample, it's going to stick itself right in between those base pairs. And it will fluoresce under a UV light. And I just so happen to have a UV illuminator up here on the board in the front of the classroom, and I'm going to project it up on the board for you guys to see and take a look at. Okay, so let me go do that for you, and I'm going to post the results up on the board here. Give me one sec. Okay, so I went ahead and projected our gel results up on the board. I went ahead and labeled them for you as well. So let's take a quick look-see. So as you can see here in the image, we have our ladder over here in the front. It's looking a little condensed. It's hard to discern the different bands in there, but that's okay. I think we can still, we get the gist. What we can clearly make out is the 200 and 100 base pair marks on the ladder. That's the important part for this particular lab, because remember, that's the sort of region that we're looking for those 35S and tubulin bands. Okay, so here we have, well, actually, let's take a look at our controls first. So down here on the bottom, we see our positive control, our GM corn. We have our tubulin gene, amplified very nicely, just below the 200 band mark on the ladder. And we have our 35S gene, and notice we have two amplicons. Interesting, right? So we have basically multiple promoters that have been incorporated into this particular sample. And if you remember that GenBag sample that we saw before that had that 489 base pair amplicon, or region, basically that might be what this amplicon is, something along those lines. So you might have the forward primer, then you have a big stretch of well, you have 35S in there, and then you have some stuff that is not 35S, and then you have the next forward primer, then you have 35S, and then the reverse, basically like that picture that I drew before. And so that might be what's going on over here. But the point is that this is known GM corn, and both primers work beautifully. For wild type corn, for our negative control over here, we have the tubulin amplifying beautifully, and we have no 35S amplification, which is exactly what we expect. Okay, so these controls tell us that we can have faith in the results of our snack food extractions. So if we take a look at our snack food extractions, pretty cool. We actually got amplification on both of our snack foods. So the cheesy corn chip looks like tubulin worked well. And we also have 2 amplicons for our 35S region. And notice they're in very much the same position as the amplicons down here. Who knows? Maybe it was the same type of GM corn. we don't know that for sure, but it's pretty interesting to see that. Then for our organic corn chip, which is a Chipotle chip, actually, we have our tubulin amplicon looking beautiful, and we have no amplification in 35S, indicating that probably there was no GM modification, or I should say genetic modification going on with our corn plants from which these Chipotle corn chips were derived. Pretty neat, pretty nifty little experiment here. So we got beautiful results. This is exactly what we should expect to see. And this experiment will work in very similar ways depending on the type of food product that you're dealing with. Now, some food products, as I said at the start of this lab in part 1, are a little bit more difficult to extract DNA from. And so that's why I say this lab is one of our hardest and most challenging labs to get good results out of. And it's all about your technique and the products that you choose to work with. And so when you come here, I challenge you to get results like these. This is beautiful, and I hope that you guys have the same when you come. Okay, so here are our results again. I just wanted to give you guys a closer look at what I was just projecting up on the board. As you can see, we have that beautiful amplification. I wanted to point out one other thing to you guys. Right over here, you'll notice these weird little ghostly looking bands, and these are primer dimer. And so those are leftover primer that's in the sample. And so what that tells you is that you actually loaded your gel correctly. So that's one of the reasons why I can tell you that, okay, yeah, there is primer dimer here. So I know that there was definitely something running through that lane there for the 35S for our organic Chipotle corn chip. And so I just wanted to call attention to that, because a lot of times that does generate some confusion. Very often we just ignore that. We'll cut it off so that you can't even see those when we post gel results online. But just wanted to point out that that's what that was. Anyway, moving on to some other student results. So this is a sample of some results that I obtained from a student lab that came here, I believe, in February of 2020. They had some pretty amazing results, and these particular students impressed me because they were able to extract DNA from clementines, which little oranges basically, and fruits tend to be one of the harder things to get DNA out of, and that's what number 1 is over here. These are clementine citrus fruits. And then we had some other results over here. I believe these may have been, I think this was actually corn chips. This number 2 was corn chips as well, and this was granola. And so basically, it is totally possible to get some good results when you guys come, but a lot of the times it does depend on the type of food product that you bring. You just have to follow the directions very carefully, but pretty neat. And the reason I wanted to project this up on the board is, if you notice in number 2 here, the students had pretty good amplification for 35S. We have clearly 2 bands here, but notice that their tubulin was a little on the lacking side, and the amplification wasn't very good for whatever reason, and that can happen. And remember, this tubulin is serving as a control in itself, and very often, if you don't get good amplification for tubulin and you are seeing very sort of sketchy amplification for 35S, then that's a control. We want to use that as such, and that might indicate that what you're seeing in the 35S amplification might just be error. But again, the 35S amplification here is pretty strong, so I'm pretty sure that the students can take confidence in the fact that there was genetic modification present. Anyway, so those are our results. Now, when we talk about GMOs, let's just take a step back before we wrap up here and put this in the context of the real world here. So we can think about GMOs in 2 ways. We can think about them in a negative context, we can think about them in a positive context. Right? So there are many benefits associated with genetic modification of organisms, and there are many possible issues with it as well. And I wanted to emphasize that genetic modification is just another type of technology. Okay, so it's a type of technology that requires research, study, planning, fact-checking, and real-world application and trials for it to actually become effective and safe for use. And so we generated a list of a lot of the pluses and minuses and the pros and cons of GMOs in industrial or sort of commercial applications. And I encourage you to do the same thing in your spare time. Come up with your own list and see if you can add to this one. But, you know, in terms of the benefits of GMOs, you know, it is a technology. It's serving to try and deal with issues that businesses, farmers, agricultural firms face in the real world. And it does so with some pretty significant and profound effects. You know, we talked about pest resistance with that Bt gene. We talked about the HT gene for the glyphosate resistance. But if you think about it, this can also be applied to plant disease like plant viruses. We talked about increasing nutritional value of crops. We talked about other environmental issues such as drought or frost. And, you know, basically improving your product. At the end of the day, you know, I think the world has certainly headed in a direction where there are a lot more genetically modified organisms being used in food. I wanted to kind of pose you guys a question. I mean, do you guys know what the top GMO foods might be? You know, and I think it's pretty obvious we're dealing with corn here quite a bit, and corn is definitely one of the big ones, but you'd be surprised to find some of the other top contenders for the use of genetically modified organisms. And so, but you know, it's pretty intuitive. I mean, like rice, soy, corn, those are obviously the big ones. Tomatoes, potatoes, big vegetable crops. You know, and interestingly, when you go to the grocery store and you try to purchase, you know, we were talking about before, you know, media and popular culture impression of genetic modification. And, you know, a lot of it is marketing. It's a big marketing game. And when you go to the grocery store, you seek out products that are labeled as non-GMO. And that's something that you're looking for. You know, at least a lot of people are. That's like a major selling point. And it turns out that it depends on the country that you're shopping in, because every country has its own particular labeling requirements regarding the terms of non-GMO or GMO and even organic, or you know, for organic products. And so in the United States, interestingly, there are no labeling requirements. And the reason for that is because it's thought that if companies were required to report on the level of GMOs that they're using in their products, that actually might hurt sales. And, you know, that's, you know, a big concern. If there's no good evidence for health concern or anything like that, you don't want to be concerned with affecting your sales, because people just simply don't want to buy your product because of the negative perception that GMOs have. It's really all about, you know, about your perception. Like organic in particular is one that I've always found interesting. So you can label a product, or companies can label their products as organic, even if they use chemicals in their fields. Pyrethrins are a class of pesticides that are derived from the chrysanthemum plants. And so because they're a naturally derived toxin, then technically the companies can label their products as organic still, even if they use pyrethrin pesticides, at least last that I had checked. So again, it's all about marketing and about your perception. So keep that in mind when you're thinking about this topic, and don't jump to conclusions and whatever social media or popular culture, however they seem to be swaying, because honestly, like I said, there are very real problems facing the world in the near future regarding the supply and demand of food. And so something must be done in order to bring food to parts of the world that struggle to meet meet the demand of food that their populations require. And genetically modified organisms allow plants to be grown in places that they never could be before, such as some of the drier parts of Africa, or you know, frost-ridden regions of the world as well. They really open up the possibilities in terms of what can be done in terms of crop plants. And so all in all, I wanted to end on this note. So if you take a look at these pictures here, think about corn for a second, just as a little thought experiment. Corn, as we know it, is this guy right over here, corn on the cob, right? It's what we see in the grocery stores and whatnot. Well, it had very humble beginnings as this little grainy grass-looking thing over here. And over many, many years of selective breeding, it was essentially created to be this, this gigantic, luscious corn on the cob that we are so used to seeing. And genetically modified organisms are simply another type of technology / process that are used in agriculture, just like selective breeding, like this, or farm machinery, right, or pesticide use or herbicide use, or harvesting equipment, irrigation technology, etc. So each one of those technologies have their own pluses and minuses and pros and cons. And GMOs are not necessarily any worse than any of those, right? They have their own pluses and minuses. In fact, some of them might be a little bit more heavy on the plus or the minus, depending on the particular genetic modification. Anyway. Thank you so much for watching. I hope you enjoyed this two-part lab series, and I encourage you to bring some exciting new snack foods to the DNA Learning Center when you guys come to visit us yourself. And I hope to see you here. I hope I'm your teacher for that. And enjoy the rest of your summer, and have a wonderful day.